Hi, my name is Lotte Bertelsen, and I'll give you a short presentation of the work performed in relation to the paper Diabetes-Induced Renal Urea Transport Alterations assessed with 3D Hyperpolarized urea. This work was performed by my colleagues and I at the MR Research Center at Aarhus University Hospital in Denmark. I will start out by telling you a bit about what hyperpolarized carbon-13 is. So, in normal MRI, we rely on a contrast agent, and particularly we rely on the imminent changes that is done to the surroundings by this specific contrast media. This is quite contrary to what we do with hyperpolarized carbon-13, because here the hyperpolarized agents will act as a source of signal themselves, rather than just modulating the signal from the protons. Also, we are able to enhance the signal of the tracer itself by more than 20,000-fold in vivo when we use hyperpolarized agents. It's also a method that doesn't rely on ionized radiation, and because it is MR, the signal that we register is specific to the chemical structures, meaning that we can use multiple components and that we can use them in a simultaneous equation and therefore acquire and see different things at the same time. However, a limiting factor of using hyperpolarized signal is the decay of the signal, because it is an artificially high signal that is created inside the hyperpolarizer, and this is lost over time and therefore limits the time window where we are able to obtain data of the metabolic and anatomical processes. It's also due to this timely dimension of the hyperpolarized MRI that we inherently have a span of 5 dimensions. The system we use at our facility is a spin lab from GE Healthcare. It's designed for clinical research, but it's also useful for animal studies. The typical tracer used in hyperpolarized carbon-13 is pyruvate. It's a very important metabolite that is central in the steps between the aerobic and the anaerobic pathway. In the anaerobic pathway, we have conversion of pyruvate to lactate, whereas in the aerobic pathway we have an entrance into the TCA cyclase with the production of CO2 and bicarbonate. In a typical experiment, we will achieve a spectrum where we can see peaks of pyruvate, bicarbonate, CO2, lactate and alanine. However, the tracer used in this study was urea, which was not metabolized, but it was used as an imaging marker of renal function, looking at the interrenal urea gradient in vivo. In this publication, we also look at animal model with diabetes. And in diabetes, there is a phenomenon which is quite specific for the kidneys, which is called PSEUDOHYPOXIA. This is where we have a pathway that is related strongly to the oxygen metabolism. The kidneys will normally extract approximately 20% of the cardiac output, so it's a quite oxygen demanding organ. And under normal conditions, it's only a small fraction of our glucose that is metabolized to the polar pathway. However, when we see hypoglycemia, as we see in diabetes, we see a flux through the polar pathway that is increased, and therefore we will have an increase of our fructose production. This also means that we see an increase in the NADH-NAD plus ratio, which eventually will lead to an increase in LDH activity, and in the same while the organ will try to maintain the constant PDH activity. All in all, we will see in these kidneys, in the diabetes, we will see an increased lactate pool, which will lead to changes in pH, and we will see occurrence of intercellular oxidative stress. So using hyperpolarized pyruvate, we were able to, in our group, to show that we have a pseudohypoxic condition already early in the diabetic rat kidney. We used the STZ-induced type 1 diabetic rat, and we saw a significant increase in the lactate level already within 3 weeks of induction of diabetes. We saw this 150% increase in the lactate pool, And we saw a lower oxygen availability in the diabetic kidney when we compared it to the healthy kidneys. We also saw that the alanine and the bicarbonate pool was unchanged. So these changes in the kidneys rely to that in the kidneys we have renal homeostasis that is determined by the active transport of water and waste products. And one of the key functions to this is the intra-renal osmotic and metabolic gradient. So as we see here and at the picture going from the cortex to the medulla, we see that the sodium and chloride gradient as well as the urea gradient is increasing into the tissues. And so based on this gradient going throughout the kidney tissue from the cortical to the medulla side, we wanted to examine a diabetic model where we see an increased oxygen consumption, which could lead to PSEUDOHYPOXIA, and we wanted to explore whether urea could be used as an imaging marker when it was hyperpolarized. So we used our STZ-induced rat, and we looked at PSEUDOHYPOXIA, and we looked at 2 weeks after induction of diabetes. And what we saw here was that there were no change in the hyperpolarized urea signal nor in the sodium gradient when we were looking at the gradient normalized to the cortical tissue. So when we look at the earlier work performed from our group, we concluded that within the first 2 to 3 weeks we see early hypertrophy, but it precludes the sodium and urea gradient changes. We see an increased demand, but we have yet to see any significant changes to the renal function. However, when we look at 4 weeks after induction of diabetes, which was the work performed in the recent paper, we see a significant change in the urea signal, and this was also supported by the regulation of the urea transporters. So it supported the increased oxygen consumption in the kidneys. So lastly, I'll try to sum up a bit about hyperpolarized renal MRI. It's a method that is based on non-ionized radiation. It's a method where the signal that we do see is quite specific to chemical structures, and therefore we can use different kinds of tracers. And in this study, we use a tracer that was not metabolized. We also see with hyperpolarization that we are able to enhance the signal more than 20,000-fold in vivo. And with previous results, we've shown that carbon-13 pyruvate may provide staging of diseases, monitoring of treatment response, and characterization of diseases, which is metabolically changing. However, in this study, we demonstrated that the non-metabolic urea signal may be an important marker for renal function because it can identify the interrenal osmolality gradient in early diabetes. And last but not least, we have recently been working with translating the things we found with hyperpolarized urea to a multipapillary system, and this shows quite great potential for introduction into human use.